lecture is hormone profiling in plant tissues, will be delivered by Andres, and hopefully he will discuss in a very elaborate way how plant hormone could be identified, then profiled, and all the details he will be discussing. So, Professor Andres.
Thank you very much. Good morning. Good morning, everyone. I would like to continue presenting the last stage detection in phytohormone isolation. The lecture title is Hormone Profiling in Plant Tissue. In this presentation, I would like to explore the critical methods and analytical techniques used in phytohormone profiling. I would like to discuss key detection parameters and address the challenges in hormone quantification. Understanding these processes is essential for advancing plant physiology, plant biology, plant biochemistry, biotechnology, and agricultural research. We know about fundamental role of phytohormones, their biosynthesis, their metabolism, the application. Despite their biological significance, these compounds are typically present extremely low concentration. We know about it, everything, but making the detection and quality challenging. Several advances analytical techniques are development in this purpose, including, of course, HPLC, GCMS, ELISA, LCMS-MS. Each of these methods, techniques, has its advances and limitations. And the choice techniques depends on the type of phytohormone being analyzed, its chemical properties, the complexity of plant matrix. A very simple reason for choosing a techniques, but it's important, is the availability of appropriate equipment. It's important to understand the key parameters that influence of accuracy and the reliability of this analysis. There are sensitivity, specificity, and quantification accuracy. Of course, the precise measurement of hormone levels requires to use internal standards, particularly stable isotope level phytohormones. This standard help correct the for sample preparation, variability, ensuring that the results are reliable and reproducible. By optimizing the parameters, this means sensitivity, specificity, and quantification accuracy, we can minimize errors and enhance the reliability of phytohormone profiling. Why are internal standards so crucial? We're analyzing plant hormones. Various factors such as sample preparation, extraction efficiency, and matrix effects can induce variability. Internal standards, of course, help correct these inconsistencies, ensuring precise and reproducible measurements. What is an internal standard? It's a compound added to the sample before extraction. It's used to account for variation in recovery efficiency during sample processing, is ion suppression in mass spectrometry, and matrix effects which may interfere with hormone detection. For phytohormones, of course, the gold standard in quantification in using isotope label internal standards. These labeled compounds behave chemically identically to the target hormones, but have a distinct mass signature, allowing for accurate correction and quantification. To achieve the highest level of accuracy in phytohormone quantification, isotope labels are widely used. What makes isotope label standards so efficient? Co-evolution with target hormones. So that means that these label hormones behave identically with native phytohormones during chromatography, ensuring accurate quantification. And the next, distinct signals in mass spectrometry. That means that due to the slightly different molecular weights, they can be easily differentiated from endogenous phytohormones in sample. I showed that some example of isotopes. Of course, by integrating this isotope in LC-MS or GC-MS workflows, we can ensure high precision, minimal matrix interference, and reproducive phytohormone quantification. Let's highlight the key advantages of using isotopes in phytohormone quantification. There are matrix efficiency corrections. This means one of the biggest challenges in hormone analysis. In complexity of plant extract, which contain various interfering compounds, isotope standards experience the same extraction and ionization condition as the target hormone, allowing for precise correction of signal, suppression of enhancement in mass spectrometry. The next, improved quantification accuracy by minimizing signal intensity. Isotope help account for simple preparation, variability, ensuring reliable quantification even in highly complex biological metrics. However, what if isotope label standards are unable? In such cases, structurally similar compounds or synthetic analogs can be used as alternative internal standards. While they may provide some level of corrections, they do not fully mimic the behavior of the target hormone, leading to reduced accuracy compared to isotope label internal standards. By utilizing proper internal standards, we can enhance the precision and the reproducibility of phytohormones analysis, making it a crucial step in plant hormone research. What's the next important in determining plant hormone concentration? We need to establish a calibration curve. Its curve is essential for converting instrumental responses, such as peak areas or intensities, into actual hormone concentration in a sample. The process of preparing a calibration curve involves several key steps. The first is stock solution, the second, serial dilution, and the third, of course, internal standard addition. By constructing a well-defined calibration curve, we can ensure that our hormone quantification is precise, reproducible, and free from significant analytical errors. The next step is to analyze, then using chromatographic techniques. I said about LC-MS, MS. Professor Shamsul presents GC-MS, MS. The goal is to construct a calibration curve that accurately relates the instrument response to no hormone concentration. Here's how we built and interpreted a calibration curve. Injection into the instrument. The calibration standards are injection into LCMS-MS or GCMS-MS, and the peak areas of the target compound and its internal standard are recorded. On the x-axis represent the no concentration of the native plant hormones. Y-axis represent the response ratio. The ratio of the native hormones peak area to the internal standards peak area. And the next, what is important, regression, of course, regression analysis. We must use specific statistical software. A linear regression model is applied to establish the relationship between concentration and response. The curve should ideally have a high correlation coefficients above 0.99, indicating strong, of course, linearity and reliability. A well-constructed calibration curve is fundamental for accurate hormone quantification. Now, what we have established the calibration curve, we can use it to determine the concentration of plant hormone in unknown sample. The process involves the following steps. Measurements of the sample. The unknown plant sample is processed and injection into LCMS-MS or GCMS system. Instrumental response, peak area or intensity is recorded. Then, comparison with the calibration curve. The response ratio of the endogenous plant hormone is compared with the established calibration curve. And the last, calculation of hormone concentration. The presented formula represent this analysis. The concentration in sample is determined using equation derived from the calibration, of course, curve. However, there are critical conditions to meet. The hormone to internal standard ratio in sample must fall within a linear range of calibration curve. The next, if the ratio falls outside the standard curve range, the quantification may be inaccurate. And the last, by carefully following these steps, we can obtain reliable, reproducible, and biological meaningful hormone quantification results. When using advanced mass spectrometry techniques such as triple quadrupole or Q-trap, there are several critical factors that must be considered to ensure accurate hormone quantification. The first is instrument resolution limitation. The next, internal standard concentration. And the third is correction factors for minimal mass differences. In the first cases, the resolution of these instruments, such as LCMS triple quad or Q-trap, is typically 1 Dalton, which may not be sufficient to distinguish interfering peaks from the natural isotope cluster of the target hormone. It can lead to false positive or overlapping signals affecting measurements accuracy. About standards concentration, internal standard concentration is said. What about the correction factors for minimal mass differences? Some isotope level standards differ from the endogenous analyte by only 1 or 2 mass unit. In some cases, a correction factor must be applied to compensate for the minor mass overlap between the labeled standard and target hormones. Here are some key quality control parameters. The first, linearity of calibration curve and limit of detection LOD and limit of quantification LOQ. LOD refers the smallest concentration of plant hormone that can be reliably detected by instruments. LOQ is the smallest concentration that can be accurately quantified, which is typical higher than the LOD. Ensuring that the method meets this criteria is essential for high confidence phytohormone profiling. There are more critical aspects must be addressed: recovery rates and reproducibility. Recovery is assessed by speaking no amount of plant hormones and internal standards into plant extract. Detected concentration are then compared to the expected values to evaluate how efficiency the hormones are extracted and measured. A high recovery rate, typically 80 to 120%, indicates a well-optimized extraction and detection methods. To ensure consistent and reliability quantification, calibration curve and sample analysis should be repeated across different days and batches. By implementing these strict quality control measures, we can ensure that hormone quantification is accurate, reproducible, and suitable for biological interpretation. As you see, table shows specific example of plant hormone quantification. By using high precision techniques, LCMS and MES or GCMS, we can accurately quantify these hormones and analyze their concentration dynamic under different physiological conditions. There are still several challenges that must be addressed to ensure accurate and reliable results. Key challenges in phytohormone detection are matrix effects, low abundance, and chemical diversity. The first, plant extracts are highly complex, containing secondary metabolites, for example, pigments, protein, sugars, and other. Then can affect signal intensity and lead to inaccurate quantification. Effective simple purification techniques, such as solid phase extraction, are essential to reduce these matrix effects. The second, of course, phytohormones are typically present at extremely low concentration, from nanomolar to picomolar levels, requiring high sensitivity detection methods. And the third, phytohormones belong to different chemical classes. For example, auxin, cytokinin, gibberellin, yasmonate, melatonin, or brasinosteroids, each requiring specific extraction and detection strategies. No single method is universally applied, making methods optimization essential for accurate quantification. Addressing these challenges requires carefully designed extraction, purification, and detection protocol to ensure that phytohormone profiling remains highly intensive, specific, and reproducible. The most common used methods are HPLC and GSME. Each technique has its strengths and limitation, and the choice between HPLC GCMS depends on the chemical properties of the hormones, sample matrix, and required sensitivity. Of course, our equipment in lab. In hormone analysis, detection is a critical step for determines the sensitivity, specificity of the instrument. Why is LC preferred over GC for plant hormone detection? Unlike GC, LC does not require derivatization. However, there are some exceptions. making it more suitable for analyzing acidic plant hormones such as auxin, abscisic acid, osmonic acid, salicylic acid. LC is highly versatile, allowing coping with multiple detection, including UV, fluorescence, and especially mass spectrometry. In my opinion, the gold standard for detector is mass spectrometry. LC-MS provides the high specificity, sensitivity, and selectivity. Mass detector allows precise quantification of phytohormones at picomolar levels, making it the most powerful detection method for plant hormone profiling. Given its higher sensitivity and broader availability, LC-MS has become the preferred method for quantifying plant hormones. Among the various detection methods available for further hormone detection, LC-MS stands out of the gold standard due to its high sensitivity. How does LC-MS work? The first step is ionization, the next mass selection, and the third step is fragmentation. In the first step, the plant extract is inducible into the mass spectrophotometer, mass spectrometer, where the molecules are ionized to create charged particles. And the next step, the second, the ions are separated based on their mass to charge ratio, MZ, using different types of mass analyzers. The last step, the fragmentation, selected ions undergo further fragmentation to produce high specific mass fingerprints, which improve selectivity. There are, of course, some types of mass analyzer in LC-MS. Triple quadruple time of flight, Q-TOF MS, and ion trap MS. Triple quadruple is the most widely used method for targeted phytohormones analysis, offering high sensitivity and quantification accuracy. Quadruple time of flight, Q-TOF MS, allows for untargeted profiling and structural identification of unknown phytohormones. Ion trap MS is used for in deep fragmentation analysis, helping to confirm hormone structures. Why is LC-MS the preferred method? No needed. I would like to summarize. No need for derivatization, unlike GCMS. High sensitivity capable of detection picomolar concentration of hormones. Superior specificity, minimizing interference from plant matrix compounds. By optimizing LC-MS parameters, we can achieve highly accurate and reproducible phytohormone quantification. In LC-MS analysis, one of the most effective techniques for improving sensitivity in phytohormone detection is multiple reaction monitoring, MRM mode, also known as selected reaction monitoring. What is MRM mode? Instead of scanning all possible ions, it focuses on pre-selected transitions, significantly reducing background noise and increasing detection specificity. It works by monitoring the fragmentation of specific precursors ion into the product ion, ensuring that only the target compound is detected. Why this mode crucial for phytohormone detection? Because it's represent high sensitivity, improve specificity, and represent simultaneous detection. How does it work? The precursor ion is selected in the first quadruple, Q1, The ion undergoes fragmentation in the second quadrupole, and the specific product ion is monitored in the third quadrupole, confirming the hormone's identity. Some, of course, examples. It's some slides present example procedure for phytohormone isolation and their chromatographic profiles. Since they were already available earlier, so I move on the next slide without the detailed discussion. It's a procedure for grinding, extraction, adding internal standard centrifuge and collection supernatant, dry dissolution in our solvent, and then, of course, using SP column and other washing, and obtain purified extract, and then injection into LCMS MS on the column. Before conclusion, the selection of an appropriate chromatographic column is a next crucial step essential for the proper separation of identification of phytohormone. Why? Next, I compare the efficiency of two different chromatographic columns used for the isolation and separation of phytohormones. The first is a kinetic EVO C18 column, and the second is standard C18 column. The key findings from this comparison are intrinsic in the following points. The first, separation efficiency for phytohormones, and the second, peak shape and in sensitivity. First, the kinetic EVO C18 column demonstrated better resolution, meaning that individual phytohormones were more directly separated from each other. The standard C18 column exhibit broader peaks, indicated lower separation efficiency and potential co-evolution of compounds, which can lead to interference in quantification. The second, since phytohormone exists in extremely low concentration, precise separation is crucial for accurate LCMS and mass quantification. For example, the EVO C18 column, due to its improved resolution, is more effective at reducing matrix interference, making it the preferred choice for plant hormone analysis. Choosing the right chromatographic column is essential for achieving high separation efficiency, reproducibility, and accurate quantification of phytohormone. What else is important? Third, in addition to selecting the appropriate LC column, maintaining high quality laboratory conditions, as I know is a good laboratory practice, is essential for obtaining accurate phytohormone measurements. The answer, of course, cleanliness, no surfactant used for cleaning floors. They can contaminate LC-MS. The second, using contamination-free laboratory consumables, such as pipette types, for example, Eppendorf tubes, or equivalent high-quality microcentrifuge types, or other plastic material involved in a sample preparation and extraction is essential for ensuring accurate results. Conclusion: analytical methods for endogenous plant hormone are essential to study the plant hormone metabolism and signalic responses. Approaches based on LC-MS have become increasingly important quantification methods. Plant extracts are extremely complex and plant hormones are present in trace amount. So the preparation procedures, which I showed yesterday, including sampling, extraction, purification, and sometimes derivatization, are very important to build high-sensitive LC-MS methods. Solution of methanol and isoprene with little water or formic acid are used to extract more plant hormones. Why, for example, acetone is suitable to strigolactons, SPE is then widely used to purify extract. Different mass spectrophotometers coupled with LC are used to develop plant hormone quantification method, in which MRM mode using triple, quadruple, or Q-trap mass has much higher sensitivity. Simultaneous quantification of phytohormone profiling and related metabolites can help understand the cross-talk within a whole plant hormone network. Simple and efficient methods determining multiple classes of plant hormone based on LC-MS is becoming possible. What about the future perspective? Improving sensitivity is an eternal pursuit and depends on the advancement of various steps from sample preparation through detection. Selective enrichment methods and novel derivatization reagent providing higher MS responses offer particular promises. The next, high throughput methods can improve the efficiency of plant hormone analysis. Increasing the level of automation of sample pretreatment is promising way. Simultaneous multi-hormone sample pretreatment via combination of multiple SPE mechanisms can present all hormonal information from one sample. Of course, we must need to excellent procedures. Improvement in MS techniques enabling simultaneous monitoring of hundreds of MRM mode transition and fast positive negative switch will make simultaneous multi-hormone detection and enable. In situ detection in MS imaging can provide spatial distribution of metabolites and their relative abundance in plants. However, it still remains far from routine of plant hormones because the endogenous level of phytohormones are so low that they cannot be detected without sample cleaning. Thank you very much. After a break, after 5 minutes, I would like to start the next lecture. Yeah, maybe more? Yeah, of course. 11:30, I will start.